Welcome to the Newsletter Out Loud. Our topic today is a holistic concept for gut health. This newsletter is a summary of a webinar previously presented by our experts Lydia Zeibich, Andrea Remesman, and Cornelia Becker. Gut health from every angle, 360-degree concept for gut health. We all know gut health is essential for livestock, but it's more than just avoiding problems. A healthy gut is a cornerstone for optimal performance, impacting both animal well-being and your bottom line. Feed additives like probiotics, prebiotics, and toxin binders can be powerful allies in promoting gut health and animal performance. They support beneficial gut bacteria and create a thriving environment. They can also help irreversibly bind harmful toxins and pathogenic microbes, providing protection. In this way, additives can help reduce the impact of the bad guys that can disrupt gut health and performance and support the good guys that aid in digestion and immune function. Indeed, a holistic approach will be the most promising one to preserve the complex nature of gut health. Microbial probiotics. Probiotics are live microorganisms that provide health benefits to the host when administered in appropriate amounts. There are three main categories of probiotics in animal nutrition: spore-forming bacteria, lactic acid bacteria, and live yeasts. Each has their strengths and weaknesses. Spore-forming bacteria are heat-stable and have the advantage in pelleted feed. These bacteria are used primarily in monogastric animals. They produce digestive enzymes to improve feed conversion and are especially known to inhibit gram-positive pathogens. Lactic acid bacteria are often used in young animals because they become active in the gut more quickly than other probiotics. These bacteria are prolific producers of lactic acid and are known to reduce gram-negative pathogens. There is one species of bacteria that combines the best of both, Bacillus coagulans. This is currently the only known probiotic that is both heat-stable and produces large amounts of lactic acid. This means that this probiotic survives the pelleting process and provides gram-positive and gram-negative bacteria inhibition, digestive enzyme production, and overall improvement of the intestinal environment. Biochem's unique strain of this exceptional probiotic is TechnoSpore, based on Bacillus coagulans DSM 32016, and has been EU registered since the end of 2020. It is also the first Bacillus coagulans registered in the EU for animal nutrition and remains the only one available for this use. TechnoSpore supports the microbial balance and reduces harmful bacteria. As such, it is effective against intestinal disorders and digestive problems. It improves protein digestion and thus supports muscle health and growth. It benefits the host's immune system and, as such, can positively affect respiratory health. TechnoSpore Bacillus coagulans DSM 32016 supports intestinal health directly and indirectly by using several modes of action, including competitive exclusion, organic acid production to improve the gut environment, and pathogen inhibition. TechnoSpore has been shown with testing both in vitro and in vivo to reduce mortality in challenged and unchallenged animals, reduce intestinal and cecal Clostridium perfringens counts, reduce pathogen-induced intestinal lesions, reduce pathogen-induced intestinal epithelial damage, improve villus height and villus crypt ratio for increased nutrient absorption, stabilize feed conversion and improve feed conversion rate in challenged and unchallenged animals, and improve body weight and control performance declines in challenged and unchallenged animals. Yeast-derived functional prebiotics. Prebiotics are non-digestible carbohydrates in food that help the growth and activity of beneficial intestinal bacteria. Although prebiotics come from a variety of foodstuffs, one source of highly functional prebiotics are lysed yeasts, which can be the entire yeast or only a part. Yeast-derived prebiotics offer more than just the classic prebiotic effects. Further, they offer pathogen binding and immune-stimulating effects and, for special yeasts, increased palatability. A commonly used yeast for prebiotic purposes is from the species Saccharomyces cerevisiae. Either whole yeasts or parts of yeast, such as yeast cell walls, are often used as functional feed materials. The main functional ingredients from the yeast cell wall are Mannan-oligosaccharides and beta-1316-glucans. The composition of yeast cell wall products is predicated by the processing and the origin of the yeast, among other factors. Mannan-oligosaccharides also have known effects on intestinal structure and function, such as significantly increased and strengthened intestinal epithelial surface area, increased villus length, resulting in improved bioavailability of nutrients in the small intestine, and stronger gut barrier for protection from pathogens. TechnoMoss positively influences gut health by supporting gut eubiosis. The high Mannan-oligosaccharide level in TechnoMoss helps to efficiently bind gram-negative pathogens, making them harmless. The beta-1316-glucans in TechnoMoss have been shown to increase the functionality of the immune system. In addition to yeast cell walls, yeast extracts provide additional benefits. One such product with both yeast extracts and cell walls is TechnoYeast, derived from the primary cultivated yeast, Cluvomyces fragilis. This unique yeast strain has a specific milky umami flavor and further delivers many functional ingredients that support feed intake, immune status, and finally gut health. TechnoYeast provides nucleic acids that are a source of nucleotides, which are important for growth, immune support, and intestinal health. These nucleotides also function to increase palatability. Additionally, TechnoYeast is a source of highly bioavailable protein and essential amino acids like lysine, threonine, and valine, as well as palatable amino acids like glutamic acid. Feed intake is a key factor in the successful rearing of young animals. In fact, there is a significant positive correlation between intestinal development, as measured by villus height, and dry matter intake in weaned piglets. The higher the feed intake, the better the intestinal development, resulting in better digestion and nutrient absorption. Field trials with TechnoYeast have demonstrated its effectiveness in promoting gut health and improved performance. TechnoYeast significantly increases feed attractivity and reduces the incidence of diarrhea, shown by improved fecal scores. Morphologically, TechnoYeast increases villus height and improves villus to crypt ratio. This results in an improved feed conversion ratio and body weight gains. In addition, TechnoYeast stimulates the immune system, including increasing the secretory IgA, which is the first line of defense against toxins and pathogens in the gut. There are numerous beneficial effects of TechnoYeast and their prebiotics, including improved feed intake, improved immune defense, reduction of gram-negative pathogens, strengthening of the gut epithelium and eubiosis, improved feed conversion ratio, improved weight gains, and enhanced animal well-being. Toxin management. While probiotics and prebiotics are often the first products that come to mind when considering feed additives to improve gut health, they are not the only ones. Another issue in the quest for gut health is toxin management. The categories of toxins relevant to the gut health of our animals are mycotoxins, the secondary metabolites of molds, and two groups of bacterial toxins, endotoxins and exotoxins. Mycotoxins can affect gut health at the cellular level. The three leading cytotoxic effects of mycotoxins are the inhibition of protein synthesis, altered gene expression, and membrane damage. All mycotoxins have lipid peroxidative effects, causing increased oxidative stress at the cellular level. Mycotoxins inhibit protein biosynthesis, resulting in a deficiency of essential proteins. These proteins include both structural proteins and functional proteins, such as enzymes. In addition, mycotoxins can alter gene expression, resulting in decreased cell performance. Although cellular damage is not readily visible, the effects can be seen in the form of disease and reduced organ performance as cellular damage accumulates. Tissues with high protein turnover and rapid growth, such as the immune system, are particularly sensitive to the effects of mycotoxins. This may lead, for example, to increased susceptibility to facultative pathogens living in the gastrointestinal tract. Another tissue greatly affected by the effects of mycotoxins is the intestinal epithelium. Only one cell layer thick, this tissue is responsible for the barrier function between the outside world, the intestinal lumen, and the inside world of the body. The intestinal epithelium is one of the fastest self-renewing tissues, renewing itself every few days to a week. This makes it very sensitive to mycotoxin exposure. Even at low mycotoxin concentrations, functional disorders can be seen as reduced feed efficiency. Other epithelial structures impacted by mycotoxin exposure are an essential part of the intestinal barrier function. These include goblet cells and tight junctions. The loss of barrier function leads to increased translocation of pathogens and other toxic substances that are always present in the gut lumen, such as bacterial endotoxins. Bacterial endotoxins, as the name implies, are toxins bound to bacteria, specifically gram-negative bacteria. In fact, 70 to 80% of the outer cell membrane of gram-negative bacteria is made up of these endotoxins. These endotoxins are also called lipopolysaccharides, or LPS, because they consist of a lipid portion and several polysaccharide chains. LPS are simply building blocks of the cell wall of gram-negative bacteria, contributing greatly to their structural integrity. They are present in every microbiome. Lipopolysaccharides are released when the cell membrane is ruptured, such as during cell death or during the proliferation of gram-negative bacteria. They exert their effects in the free form. There are several other factors that determine how damaging LPS can be. One is location. Do they remain in the intestinal lumen, or can they cross the gastrointestinal barrier to enter the organism? Another is number, the level of exposure to LPS. Third is species. Some species, such as poultry, are less sensitive to LPS than others, such as ruminants and pigs. When the intestinal epithelium is healthy and has good integrity, endotoxin uptake is controlled, resulting in a limited amount of endotoxin in the organism. These endotoxins are transported to the liver, where they are presented to the immune system. In this way, the immune system is regularly trained for the markers of gram-negative bacterial proliferation. However, when gut integrity is compromised, as in the case of mycotoxin exposure or heat stress, this controlled uptake of endotoxin becomes uncontrolled. As a result, the neutralizing capacity of the liver is overwhelmed and the endotoxin spreads systemically. Endotoxins block certain receptors, leading to an inflammatory response. The more endotoxins, the more severe the response. One such inflammatory response is porcine ear necrosis. This disease occurs mainly in weaning piglets. A 50-day trial was performed with 408 healthy weaned piglets to determine the effects of supplemented a toxin binder on endotoxin-associated disorders. Piglets were placed in two groups and were fed either a basal diet or the basal diet with a toxin binder, B.I.O.Tox activate, which was selected because it binds all relevant mycotoxins. Groups were assessed for growth performance as well as the prevalence of endotoxin-associated disorders such as ear necrosis, and the feed was tested for the presence of mycotoxins. The amount of ear necrosis was determined by visual assessment and assigning a grade depending on the amount of necrosis present penwise, using the following scale: grade 0, no piglets in the pen are affected. Grade 1, less than 30% of the piglets in the pen are affected. Grade 2, more than 30% of the piglets in the pen are affected. And grade 3, all of the piglets in the pen are affected. Piglets supplemented with B.I.O.Tox activate had reduced incidence of ear necrosis compared to the control group. Moreover, when compared to the control group, severe cases were significantly reduced in the toxin binder group. Analysis of the diet demonstrated that the only mycotoxin present was DON at a level of 60 to 100 parts per billion. Adding B.I.O.Tox activate to the feed also resulted in reduced losses, a numerically improved feed conversion ratio, as well as better uniformity between the pens. These results imply improved gastrointestinal integrity with the addition of a toxin binder. The visible effects of endotoxins are only the tip of the iceberg. So much more damage is occurring beneath the surface. Good endotoxin management aims to avoid as many factors as possible that compromise the integrity, such as avoiding specific pathogens, removing harmful mycotoxins, and reducing heat stress. Endotoxin-related disorders can be reduced with the smart selection of feed additives embedded in a holistic feed and management concept. Bacterial exotoxins. Whereas endotoxins are structural parts of gram-negative bacteria, exotoxins are direct weapons. They are proteins excreted by certain bacterial strains with possible necrotic or hemolytic effects. The most relevant are the exotoxins produced by Clostridium and those produced by Salmonella species or E. coli species. These bacteria are present in all 3 livestock animal groups as a normal part of the gut microbial community. So when do the presence of these potential pathogens become a problem? The development of necrotic enteritis is a good illustration of how these exotoxin-associated diseases develop and how they can be prevented. Clostridium perfringens produces toxins when a certain population density is reached. This critical mass can be reached due to the dysbiosis and reduced immune status, but also due to physical changes in the gut, such as mycotoxin-induced leaky gut or reduced intestinal resilience. Mycotoxins disrupt gut integrity and may increase the virulence of certain pathogens, paving the way for increased endotoxin stress and indirectly promoting exotoxin formation. They can be neutralized by specific solutions such as highly effective surface-activated binding agents. Efficient mycotoxin management is the basis for holistic toxin management. Neither endotoxins nor exotoxins can be controlled by a one-size-fits-all product. For the most promising outcome, a smart approach to feed additives is required, in addition to appropriate feed formulation and management. For the most promising outcome, a smart approach to feed additives is required, in addition to appropriate feed formulation and management. 360-degree gut health concept. Several factors can disrupt gut health, including harmful microbes, toxins, and even some medications. To address these challenges, we need a holistic approach. This includes smart medical interventions, such as the balanced use of antibiotics and strategic vaccination programs that can help maintain a healthy gut. Effective management practices, such as proper hygiene and housing conditions that play a critical role. Optimal feed formulations that give proper nutrition and providing essential nutrients and supporting a healthy gut microbiome. Feed additives can make a significant contribution to a healthy gut. However, there is no single solution to all problems. Only a holistic 300-degree approach from all angles will efficiently lead to a robust gut health and improved overall animal performance. I'm Dr. Melinda Culver for Biochem, and thank you for listening to the newsletter out loud. If you would like to learn more about this topic or any of the products I mentioned, please visit our website at biochem.net.